Yes. So thank you for the invitation to start the year. And yes, I will share a bit of our work on the blood circulation modeling, and especially in the liver across different scales of the organ and the whole body. So let's begin with fundamentals. So here are the Navier-Stokes equations for fluids. You can couple this to the solid wall within a blood vessel, for example, to simulate blood flow or airflow in vessels or conduits. And sometimes it's too costly to do simulations in 3D. So we can integrate these equations across on a cross-sectional area of a blood vessel to arrive to so-called 1D equations, but with the same physics. So inertia, balancing the pressure forces and the viscous terms, so the pressure loss due to viscosity. And also conservation of mass that you find here. And then we can also further integrate these equations along a blood vessel. And we arrive at ODEs this time, where the same physics holds, of course. So we find, for example, this equation for the inductance that represents inertia of blood, capacitance for the taking into account the conservation of mass when there is wall elasticity, and loss of pressure due to the resistance to the flow due to the fact that blood is viscous. And so here you see an analogy between blood flow and electric systems, where pressure is like voltage and flow rate is like current. And more we combine these equations, either for fluid or also for transport. We can have the same kind of scheme of different models for transport. So we couple these equations with the machine learning models, typically when we don't have the equations that model a certain process. So then how do we choose which model we use for biofluid or transport? Well, it really depends on the biomedical question that we are trying to solve. So typically for device design, we use 3D simulations or inhaled particle. But to understand wave propagation phenomena from the heart, usually 1D methods are favored. And then if we want to understand the interaction between an organ, such as the liver and the rest of the circulation, like the heart, for example, or other organ, we use this electric analogy that permits us to easily model these different components of our circulatory system. We also use these reduced models in large networks of vessels at the macrocirculatory level, and I will talk about it more in a minute. So all this is in the context of like a new word, a pop-up word that maybe you have heard a lot about. So I thought it's good to label a bit what we mean by digital twin of a patient. And this is part of the work of a European consortium called EDIT, which aims at gathering together all the human virtual twins developed within Europe. So the digital twin of a patient is a model that represents, so here, the blood circulation of a patient, and that is at a level of fidelity that is, as I showed, coherent with the biomedical question at hand, and which is tuned from data. And the data, we have it at a given state. And this allows us to either provide new information of a given state that typically is difficult to measure, or it enables to predict another patient state, such as during a surgery. So I will give a more concrete example afterwards. But in any case, we always start from some geometrical information, so from some imaging data, and the modality depends on the scale at which we are studying. And from the imaging data, we can segment some vessels and then perform numerical simulation in these vessels. So there are several challenges, especially if we want to do subject-specific parameterization. We have the first challenge that hemodynamics data, so typically pressure and flows, are usually not done where we need to impose boundary conditions. So typically at inlet or outlet of vessels. They're always uncertain in their acquisition, and they're often too few to, well, I would say always too few to identify all the parameters. And also sometimes they're not taken simultaneously. So we may have pressures that are measured in the cath lab, and then flow rate or imaging data that are taken in the MRI suite. So they may even be taken at different heart rates, so at different states of the overall system. And then from a numerical point of view, the implementation computational complexity for to identify the parameters of the model. So here the 0D resistances, capacitances, and inductances. So this parameter identification need to be like coherent with how much data we have to retrieve these parameters. So we'll touch upon that more at the end of my talk. So we've been performing flow and transport modeling for different applications, but especially, for example, for surgical planning or development of a disease. For this, you really need to have a predictive model, and this, in fact, let's say. Requires that we couple 3D models typically with, so 3D model of the region of interest with the rest of the system. So typically the rest of the circulation. So of course we cannot model the rest of the circulation in 3D. So this is why we couple 3D models with these so-called 0D, so ODs models. And then we can accelerate simulations if we have to do a lot of simulations such as for medical device design, for example. And for this, we can use POD or other ways to reduce the simulation costs. And also, we can use machine learning to go from an ill-posed problem to a well-posed problem. So basically to use machine learning as a way to provide some prior information, which then makes the problem of identification well-posed. And then finally, yes, we are using more and more machine learning methods, for example, to predict something for which we have the equation for a given state, but we don't know yet the equations that predict the process, describe the process of the evolution of a disease. So for example, in aneurysm growth, so when you have like this enlargement of the vessel wall in the aorta, sometimes it's increasing a lot over time and sometimes not. And so we can describe the state of an aneurysm with geometrical description, with fluid mechanics descriptions, but not yet the growth and remodeling. And so sometimes we combine so PDEs with machine learning for that purpose. Okay, so I will use some of these methods, let's say, in the context of liver, which is the subject of today. So liver is the main blood detoxifying organ. So it's a very important organ right below the heart, in fact, that receives the blood from different digestive system. And therefore it has like a double perfusion. So it receives the what we call the artery blood, so blood that is oxygenated to sustain the organ, but it also receives blood, so venous blood coming from the stomach, the intestine, etc., that it has to detoxify. And in fact, liver is the main blood detoxifying organ. And this detoxification takes place in liver functional units. So here we are zooming inside the liver. After many branches of these major vessels that are perfusing the liver, small vessels are actually perfusing the cells of the liver. And this is done in an architecture that is quite remarkable. So here we are talking about a liver lobule that has this kind of hexagonal shape, or at least is often described like this. Blood goes from the what's called the portal triad, so where this double perfusion enters, towards the central vein through capillaries, very small vessels, where the exchanges with the liver cells, so the hepatocytes, take place. And these processes are very important for the function of the liver. So overall, this means that both perfusion of the organ and the function, let's say the cellular function and all the exchanges between the vessels and the cells are really key for a well-functioning organ. This is why it's like a multi-scale, really a multi-scale organ, so to speak. So I will first start with the macro scale, so the lobule level, where we've been studying hemodynamics model and transport. So at that level, so on the histological level, we start from the geometry, so the network of the blood vessels. And in fact, if we take a real network, it's really challenging to know which boundary conditions to put at the border of these all these tiny vessels. This is why very often we work with like a statistically representative lobule that is generated according to statistics that have been measured in histological reconstruction. And this work has been done lots by Stefan Hömme, who is, I've seen, is online. So once we have the geometry, then how do we model blood flow? So we start with this resistance, so Poiseuille resistance, which is a very simple law relating linearly a pressure difference to the flow rate via the resistance to flow. This resistance is proportional to the viscosity of the blood, the length of the vessel, and inversely proportional to the radius to the power 4 of that vessel. And then we apply conservation of mass at every node of the network. And then we need to prescribe some closing condition of the system. So here it's a big algebraic system. So either pressure or flow. And this viscosity actually is a bit complex at that level. So the resistance to flow is complex because in fact, blood is not like a classical fluid. It contains red blood cells and other things. So how much red blood cells are inside the volume of fluid is called the hematocrit. So it's a concentration. And in fact, the change of blood viscosity due to the hematocrit has been described as the Ferris-Lindquist effect quite some time ago. So we can relate the viscosity to the hematocrit and the radius via a certain known and quite complex function. And we've shown that this rheology is needed. So this rheological model is needed to reproduce average velocities in these small capillaries. And here on the left, I put a graph of that shows how the viscosity is changing when the diameter is increasing of a vessel. So you see the nonlinear effects of this law. Now there is another effect due to red blood cells, which is the partition of the red blood cells at a bifurcation. This is called plasma skimming. This partition is not even in the sense that it's not proportional to the split of the flow rate between the different branches. And so this has been observed experimentally. And so here we'll consider the fraction of blood flow in bifurcations going from a mother branch to two daughter branches, as well as the fractional red blood cell flux. And so this effect is also changing the hematocrit. This has been described by a nonlinear law by Price et al. Or by a linear law by Gould and Lillinger more recently. So the advantage of the linear law is that, in fact, it can easily handle trifurcations or even more complex intersection. But when compared to experimental data, which are the dotted blue dots or green dots here, of the fractional red blood cell flux versus fractional blood flow, we see that, okay, when we have no plasma skimming, of course, we have just a simple line here. But the linear law does not really capture well what is observed experimentally, contrarily to the nonlinear law. The problem of this nonlinear law, nevertheless, is that it cannot easily be extended beyond a bifurcation. And we sometimes see that in networks. So that's why we started to look into these models and to see if we take into account a number of parameters that we found in the literature for different typical bifurcations that are in the liver or in other networks in different species. What we have found basically is what you find on the right. So we model all these different cases. It's not very important to understand how these different cases differ. But what you can see is that when you compare no plasma skimming in red to the linear models in orange and green and the nonlinear law, for each case, in fact, what matters is to see if the repartition between the two branches that of the flux that you see with a little bar here each time, if it's closer to 0.5 or not. And so what we see is that each time the nonlinear law actually first has more influence compared to no plasma skimming than a linear law. And also it tends to be closer to 0.5. So it's tending to homogenize the flow in the system in simple bifurcation. Then what was interesting in this, a little bit puzzling at the beginning, but interesting finally, in this nonlinear model, is that sometimes we found two different states in the system. So here I'm plotting different graphs with in each graph the radius, increasing radius of the one daughter branch and increasing radius of the second daughter branch. And you can see this phase diagram where depending on the radius of the mother branch, we have different phase diagrams. So we have two states that are found in green versus one state in blue. And this phase diagram evolves depending on the mother branch diameter. So basically, there are two regions in larger capillaries, which have similar daughter branches. We have these two states happening. And in smaller capillaries, like we have two states with two different hematocrit level. But contrarily to the first zone, here we have an oscillation between one branch that has 0 hematocrits and the other one that has the rest of the hematocrits. So it's like flipping sides, if you want. And in fact, these two states are consistent with experimental observations and also modeling in some other papers. So this was really interesting from a modeling point of view. So then we started to look also in the heterogeneity of hemodynamics in the whole lobule. So here it's a 2D projection from above for the pressure, the flow, the hematocrits, and the washer stress. Because in fact, in a number of mathematical models of metabolism of the liver, they use like a single blood vessel that represents the entire lobule. So they say, okay, we are going to, instead of considering this complexity, we are going to only use like a single representative vessel going from the inflow to the outflow of the lobule. And in fact, we see that this may not be a good model to do that, because we can see that the flow rate in particular is quite inhomogeneous throughout the lobule. So what I've shown on a simple bifurcation, we have found it also in a whole lobule. That is that the plasma skimming, so the effect of the red blood cells is really important to take into account, as it's homogenizing the flow in the system. And we found again that the linear plasma skimming law has less effects compared to the nonlinear law, with the baseline as being no plasma skimming. But then what really matters, in fact, is to see between healthy and disease states how these parameters change. And a disease state can be represented typically by like a geometrical variation. So this is why we compared like a whole lobule with constant vessels to like a Gaussian distribution of vessels, and to a geometry where there is dilation of vessels close to the central vein, so close to this zone. which has been observed, for example, in COVID-19. And here, actually, we found that this geometrical variation has more impact than the rheological variation that I talked about before. So more impact on the pressure, the flow rate shown here, but also the velocities. So this is why for the rest of our studies, we considered that, okay, from a rheological point of view, we are going to keep the first effect, the Farris-Lindquist effect, but we will discard the plasma skimming effect when we study different architectures. So for the flow point of view, and also for what is transported by the blood. So to model compounds that are injected in the blood. So compounds that can be drugs, for example, or contrast agents that are injected in the blood for medical imaging, typically, or concentration of metabolics. So, for example, when some colleagues study ammonia detoxification by the liver. So once we have, in terms of modeling, once we have the velocity from the hemodynamic study, we can study transport. So here, pure advection in this network, and with some reaction term, because what is transported in the blood is partially exchanged with the hepatocytes, so with the neighboring cells, sorry. So this here is modeled by a active reaction term of the Michaelis-Menten form. And actually, when studying either metabolism or contrast agent spread throughout the liver, the concentration that we want to study at the inlet of the lobule can be of different shapes in time. So it can have a profile that is quite peaked, or being a large Gaussian. So you can imagine that from a numerical point of view, it's not going to be the same it's going to be more complex if you have to deal with like a sharpened input function. So we wanted to have a numerical scheme that can handle multiple multifurcation, and that handles these several types of input function shapes. So here we have essentially a convection reaction model that is that we describe actually in 1D, like in these different tubes, with like a certain prescribed inlet condition. So for this, the discretization, we use an explicit finite volume scheme, which is quite classical, with an upwind part for the flux, but for which we provided extension of flux of well-known flux limiters, because in the literature, what was existing was like flux limiter for like 1D problem. So, but here the problem is that when you have something transported in converging bifurcation, when you are at this node here, in fact, the flux emitters needs information of the upstream node. And in a network, it's not that easy to know which information you are going to provide, because the information is coming from different blood vessels. So here we proposed a flux, like a flux limiter, that conserves mass, and it worked quite well. So it works quite well for a number of common flux limiters on simple as well as complex networks. And here it really handles multiplication, and we could show that mathematically it's preserving positivity, and it's conserving mass by design. So here you see the L2 error for like a simple bifurcation, so converging bifurcation versus bifurcating bifurcation for, yes, 5 different slope limiters. So the behavior is roughly the same for these different slope limiters. And then, so we wanted to see how it's handling the different input functions. So a bolus, or a shape like this, which is closer to what you find in medical imaging, or like a sharp rectangle or a large Gaussian. So you note that here you have, you are in the time scale of hours, whereas here it's more like seconds. And so what you see is that, okay, when you have a large Gaussian, it doesn't really matter which numerical scheme you use, but if you use like a sharper Gaussian or this typical imaging, medical imaging input curve, you see more of a difference in the transported curve. So we concluded that for sharp inputs, you really need this flux limiter to reduce the numerical diffusion. And also we looked at different types of reactions. So either like a simple convection, like where there is no decay in time of the profile, so not many exchanges between the vessel and the cells. Or we can observe for like a linear regime of this reaction term, like a exponential decay or linear decay. So you have different regimes, and this is good because you can compare in these cases with analytical solution when you're in a simple tube. So we could, verify our numerical schemes that way. And then when we looked at something that is called the mean transit time, so how much time it takes for this input curve to pass through the entire network. So here we could see that, so it's an integrated measure of what's going on. In fact, there was not much difference between the different geometries. So we were a bit disappointed because you can see some differences in the transport of this curve between constant radii, healthy and dilated. But this is really at the if you look at the whole temporal curve, whereas if you look on average, like at this mean transit time, you don't find any difference. So this means we need to find new biomarkers, new signature of how geometry impacts transport, and this is the subject of a current PhD in the team. All right, so this is what we did for the lobule level, but then, so this is when you go from like a real lobule to like multiple lobules to a whole liver organ, you can imagine that you have then many lobules in parallel. Well, it's not very known if they are only all in parallel or partly in parallel and in series. So this is something that we are investigating in some projects. But we know that when the disease progresses from being local and happening at the liver lobule, at some point, clinicians can see some effects at the global liver organ. So this is why we are also looking at the scale and at how the liver is connected to even the entire blood circulation. And this part is now more connected to what Marie said at the beginning, so to surgery. So to treat some cancers or other chronic diseases, surgeons perform, for example, a partial hepatectomy. So they remove parts of the liver, and there is a certain risk associated with that. So the risk that is associated with either partial liver resection or transplantation of a small liver is that at the end of both of these procedures, you have a much smaller liver than at the beginning. So, you know, it's a little bit when you go to large highways on vacation, and all of a sudden they are doing rebuilding, and you have three roads blocked, 3 lines blocked. You have just 1 line remaining. Well, there is a high friction of flow of cars going through. Well, here it's the same. So there is actually a large tranche of pressure in happening in the liver in that case, because the liver is still getting all the blood from the body that it needs to detoxify, even if it's like a small piece remaining. And depending on the quality of the liver, it's really difficult for the surgeons to appreciate, like to predict what's going to happen for a certain liver volume for a given patient. And this way we started to work together. Yeah, part of the problem is this is the creation of a too high pressure at the entrance or inside, in fact, the liver. But what they can measure is at the entrance of the liver. So the recommendation, the European recommendation for liver surgery changed in 2018 and said, okay, now you can operate if there is portal hypertension, but there can be an intermediate risk or a too high risk to have the surgery, depending on how much liver you're going to take away and depending on the quality of the liver. The problem is that it's not so much the pre-surgery pressure, which is a problem, but the pressure at the end of the surgery. So the pressure increase that is due to the surgery itself on top of a diseased liver. And to predict this, in fact, surgeons don't have any tools for this. And this is in this context that we thought we could use a model of the hemodynamics in the entire circulation. So with the heart, the liver, but also the digestive systems and the rest of the organs, the lungs. So we made an analogy, an electrical analogy of this whole circulation with a zoom on the liver. I don't know if you see that because I see the videos, but we can zoom on the different parts of the liver, and we can perform like a virtual surgery to predict the increase of pressure and the decrease of flow in the liver. So we did the first study, and this is what I will talk to you about now briefly. So we have this model, which is actually an ODE system with many parameters to fix, with like a double perfusion part of the liver for the right side of the liver and one for the left side of the liver. So in fact, it's a nonlinear algebra differential system, which you can rewrite like this. So as a semi-implicit algebra differential system of in this one. So writing it like this ensures existence of the solution. And we are actually solving this coupled system of equation with a IDA, which is provided by sundials. So in time, it's a BDF scheme, time scheme, but that is variable in order, like from 1 to 5, according to the time step automatically. So this is built in IDA. And for the nonlinear part, it's using a Newtonian method. So as you see here, so we have actually pretty few patient data to parameterize the whole model. So we know the total liver volume, how much will be resected, and then we have a couple of pressures and flows in the system. So what we do is we fix some of the parameters, and the rest we adapt it with a genetic algorithm to fit the remaining parameters based on these measurements. Then we can simulate the partial hepatectomy for each patient individually. And then we looked at the pressure at the entrance of the liver and the change of pressure across the liver, which are the two quantities that the surgeons care really about. And we had to change our model on the fly because we saw that what mattered also is what's happening during the operation in terms of blood loss, infusion of fluids, and change of systemic vascular resistance due to the surgeon's action on the vessels. So in the first study, we compared the output of these two types of simulations for the pressure at the entrance of the liver and the gradient, what they call the gradient of pressure across the liver, with the measurement on the left. And so you see that statistically, it's quite good for now, even if we still have some progress to make if we want to use it as a predictive tool. So here are the results comparing the computed versus measured pressure at the entrance of the liver. So you see a quite nice correlation. And especially if we look at so for this first group of patients for which the underlying liver was not too diseased, there was a known clinical cutoff value of 21 millimeter mercury, above which it's like there is a higher mortality after the surgery. And you see that we can pretty well predict which patients are above or under this value. Another way to look at this simulation is to look at the difference between the simulated and measured data versus the average. So this is called a Bland-Allman graph. And you can see that all the dots are within the tolerances that our clinical experts said are the important range to manage. Of note, in particular, there are 4 patients for which we predicted quite high pressure gradients. And these are the ones that unfortunately suffered from what politely the clinicians call a PHLF, so postoperative liver failure, which means that unfortunately the patient died. So there is something that is a bit under the hood here. It's the calibration process and how uncertain our predictions are. And so here it's how it works for now. So we have these measurements. On one hand, we have literature data to fix some parameters, and then we do a calibration step to adjust the parameters as much as we can to the measurements. I mean, so that the output of the model is close to the measurements. This gives us the rest of the parameters. And then we can do the simulation step and look at some output of the model Y. So here we started to do a sensitivity analysis based on Sobol indices. So this it's a mathematical tool to reveal like which parameters among the ones estimated from measurements and literature assumptions are the most impactful on the output. So which parameters can we consider more constant without losing any accuracy, and which ones do we really need to focus on? So so the Sobol indices method, so the S here, it's a global sensitivity analysis, and it's based on the variance, the variance of the output given certain inputs. So here parameters, and we will look at two things. The first, what is called the first order Sobol index, which describes the direct influence of a specific input, so a specific parameter over a certain output, so typically a pressure. And the total Sobol index, which describes the direct influence of a specific parameter, but also the interactions with the other input parameters over this specific output, so pressure at the entrance of the lever, for example. So how did we do that? So we, from this first study that we did, where we fitted the parameters, we could have empirical distributions of different parameters, so resistances and elastances of the heart. Also the extent of hepatectomy. So we had the distribution over this first cohort. And then we could, with the spread of each parameter, we could compute we could do the sensitivity analysis. So we could provide through the model, like the distribution of each output of the model. Okay, so for example, the distribution of the pressure in the liver at the entrance of the liver before the surgery, when we vary all these input parameters and we compare them to measurements in orange. So this allowed us to do this sensitivity analysis. So for each output, to look at how close to one the Sobol index is for each input parameter. But when we did this process, in fact, we saw that there were some output of the models that were no longer physiological, because when you vary all the parameters in the parameter space, there is no way to impose that the output will be physiological. So hence we had to go back and think about how to deal with this problem. So what we did is we defined with our clinical colleagues ranges of clinical output that would be acceptable. And we filtered all of our results, keeping only all the varying parameters as inputs and the outputs we got that are physiological. With these trusted information, let's say, we used the polynomial Chaos expansion methods to build a reduced model that is mapping these input parameters to the outputs. So here we no longer use the ODE model, but like a polynomial Chaos expansion method. But that is built on the trusted values. And it works well, and it's very efficient. And now, in fact, all our outputs are within bounds that are clinically acceptable. So this allowed us to generate actually a virtual population that we shared with everyone that wants to access it. So on these, because of using a polynomial chaos expansion for the reduced model, it's very easy to compute actually the suball indices from that. So we could redo this analysis. And here are the results. So for the sensitivity after partial hepatectomy, you have a heat map like this with the main clinical outputs that the clinicians care about and the main parameters that we tune out of these measurements. And what you see actually in the first two rows is that the two pressures that the clinician have a difficulty to anticipate, they are indeed related to a lot of input parameters. So this makes sense that for them, it's a bit difficult to predict in their head what's going on, because it's true that among all the pressures and flows that we look at in the system, they are the ones that are, the mostly that are related to a large number of parameters with a high sensitivity. And then there are other factors that were interesting. But then this raises the question of, okay, so if they're related to all these parameters, if they are quite sensitive to all these parameters, how can we identify all these parameters with the preoperative measurements? And so this is what this sensitivity analysis on the preoperative state allowed us to do. And in fact, we can see that most of these parameters are related to measurements that the clinicians perform. And there is one parameter actually that is not related to any measurement. It's an elastance parameter on the right side of the heart. But in fact, we see that it's so it's hard to estimate it. But on the other hand, it doesn't have a significant impact on the output that we're looking at. So it doesn't matter. So now we are continuing this uncertainty quantification analysis, but not on the whole population level, but for each patient. And we are doing an identifiability analysis to see which other measurements we can perform in a new clinical study with our clinical partners.
Irène, maybe you could try to conclude in a few minutes so that we can have some questions.
Yeah, exactly. So I'm almost at the conclusion here. So just at the end thought, let's say, because the surgeons are very creative, so they see that we can do some things with a digital twin. But the last question they ask us is, can you then predict the complexity of the surgery? So if I give you preoperative scans, can you predict the intraoperative complexity as they note it at the end of the surgery? But for this, like, I don't know, we don't know any mathematical equation that represents that. So in this context, or for this kind of question, I think there is no other way than using machine learning. So that's why we started to use deep learning methods to automatically segment liver, vessels, tumors from single scans, and then extract relevant biomarkers from this segmentation and predict then the liver resection complexity. And actually it works quite well. So I think the future will be to mix these kind of approaches to be more more robust in what we predict. Okay, so if I conclude, so at the micro scale, we've seen that in fact, there are different mathematical models that describes quite nicely what's going on at the bifurcation levels. But in fact, taking into account geometrical variation as for diseases, we don't necessarily need very refined biological models to see what's going on for blood flow and transported material. We've also seen that from a numerical point of view, if we look at transported compounds, we really need to be careful about using proper numerical schemes. But that nevertheless, in fact, in all what I've showed you, reduced models are really enough compared to 3D models to help to anticipate surgical risk. And then when we move on more to the surgical aspect and the patient-specific analysis, yes, there is still quite to do to understand the identifiability of parameters through sensitivity analysis. And then finally, I think we can combine, yes, machine learning and physical modeling to answer some complex questions. So thank you for your attention, and I'm happy to take any questions.